What is spermiogenesis? Remember to describe spermiogenesis under these headings. Spermiogenesis is again a morphological process by which a round spermatid or definitive spermatocyte is converted into an elongated spermatozoa. What are the morphological changes and where these changes occur? Spermiogenesis also occurs in seminiferous tubules. What happens here is the round nucleus of the mature spermatid or definitive spermatocyte will get converted to form the head of the sperm. The head of the sperm is covered by an acrosomal cap. This acrosomal cap is derived from the Golgi apparatus. Then you have the centrosome in the spermatid, which gives rise to proximal and distal centrioles. The proximal centriole will form the neck or is present in the neck, and this will give rise to axial filaments. The distal centriole will form the annulus through which these axial filaments pass. Then we have the mitochondria. The mitochondria forms a sheath in the tail of the spermatozoa, generating power for spermatozoa. The cell membrane of the spermatid retains as the cell membrane of the spermatozoa. So these are few changes which result in the formation of an elongated spermatozoa. Now, the mature spermatozoa measures about 50 microns and is divided into head, neck and tail. Tail is further divided into the middle piece, principal piece and end piece. The head measures about 4 to 6 microns. Then the neck is the shortest portion, measuring about 0.3 to 1 micron. Then rest of the length of the spermatozoa is formed of tail only, about 40 to 45 microns. Of this, the initial portion is called as middle piece, which contains mitochondrial sheath. This is the powerhouse of spermatozoa. In the recent INICET exam, there was a question on which part of the spermatozoa helps in generation of power. So that is the middle piece. The middle piece is followed by the presence of the principal piece. This principal piece is responsible for the motility of the spermatozoa. This is followed by the end piece without any sheath. This is regarding the mature spermatozoal structure. So this is how the prenatal maturation occurs. Now let's talk about the process of postnatal maturation. What happens in postnatal maturation is, once the female child attains puberty and starts having menstrual cycle, the first few menstrual cycles are anovulatory. Ovulation does not occur. Then occurs ovulatory cycles, where each month one oocyte will be released into the peritoneal cavity, right? This process is called as ovulation. Ovulation roughly occurs at about 14th day of a 28-day menstrual cycle, in the middle of the menstrual cycle, because of a surge in the level of LH or luteinizing hormone. Now just before this ovulation, the first meiotic division gets completed. Remember, before birth, the primary oocytes are arrested in prophase 1 of meiosis 1. Now this prophase 1 becomes active again, right, just before ovulation. So the primary oocyte completes its first meiotic division, forms the secondary oocyte, giving rise to two cells, one with large amount of cytoplasm, which is called as secondary oocyte, and the second one is called as polar body. So polar body receives nuclear material, secondary oocyte relieves nuclear material, but secondary oocyte relieves almost all the cytoplasmic material and polar body receives none of it, right? So the cytoplasmic division is unequal here compared to the equal cell division that occurs in spermatogenesis, right? So this is meiosis 1. Now the secondary oocyte that is formed should enter second meiotic division. Very good. Now this second meiotic division gets arrested again. So oogenesis is getting arrested twice, one before birth and one after ovulation. But now the secondary oocyte is getting arrested in the stage of metaphase, right, metaphase 2. This metaphase 2 will be complete only with the entry of spermatozoa. So, if there has to be one successful cycle of oogenesis where there is complete maturation of secondary oocyte into zygote, sperm has to enter it. This is one more peculiarity of oogenesis. So, what happens in second meiotic division? The secondary oocyte will further undergo division, giving rise to your zygote, and then we have the second polar body. The polar body, which is formed during the completion of first meiotic division, undergoes further meiotic division, giving rise to two polar bodies. So, in the end of oogenesis, we have one zygote, right? That is after the completion of the fertilization, right? Once the spermatozoa enters, so we have the fertilized zygote, we have three polar bodies. Even this has been asked in your entrance exam, okay?